Hello everyone, myself Deepa Singh, working as assistant professor in Gyan, Vihar School of Pharmacy, SGVU, Jaipur. Today we discuss about viability and bioequivalence. In previous lecture, we covered already small part of viability, what is viability and their definition. So today we discuss about complete description or about the viability and the bioequivalence both parameter. Means what is viability and what is bioequivalence and the difference between these viability and bioequivalence, and there are certain parameters of this viability and bioequivalence, including pharmacodynamic, pharmacokinetic parameter, and study of this viability and bioequivalence all we covered in this lecture. So this is the topic of biopharmaceutics and pharmacokinetic subject 6th semester. Before starting this topic or this viability and bioequivalence, you should know about basic object or learning object. In learning object, you should know about introduction of the viability and bioequivalence. Means what is viability and what is bioequivalence, the definition, the description part, and how to calculate viability and bioequivalence. Maybe different methods or different formula of this viability and bioequivalence. So in this, you also know about the calculation part of viability and bioequivalence. Next is component of viability. Means what are the components which is required for viability and bioequivalence. Measurement of viability. You can measure the viability by two methods, that is direct and indirect. And this method is also known as pharmacokinetic and pharmacodynamic methods. So you also know about these two methods, or direct or indirect methods in this topic. Next content of this topic is objective, introduction part, definition, and objective of viability and bioequivalence both, significance, components, measurement of viability in this method, direct and indirect method, conclusion. In objective part, to study the various objectives of viability and to know the significance of viability. Means what are the objective of the study viability and bioequivalence. Means you can say that you can study the object or various object of viability by performing this viability study, or the object of viability study with the significance and significance of viability, and to study various measurement parameters used in measurement of viability. Which parameter you used and how you perform this viability, and the measurement parameter means including method as well as formula. You should know all this in objective of viability. Next, in introduction part. In introduction part, viability denoted as a defined as F, or maybe denoted by F, and when you calculate the percentage of viability, then you write on percentage symbol, that is qualities that qualifies the proportion of a drug which absorbs and available to produce a systemic effect. Means it's the amount of a drug or quantity of a drug that produces in a system, produce the systemic effect or goes in your systemic circulation to show the particular desired therapeutic effect or available in the systemic circulation. Next, in this, viability is a fundamental property of pharmaceutical product for a given route of administration. Means you can also say that it's a fundamental property when given a particular route of administration, or according to route of administration. We should be know and show the producible for the all drug product intended to produce a systemic effect. Means all type of drug are required to produce a suitable and desired effect for systemic circulation or in the systemic circulation. Means this viability is denoted by F, and this is the amount of drug or quantity of a drug that required to show the particular desired effect, and the drug which is in a systemic circulation. And you can also say that this is the fundamental property of pharmaceutical drug, and this is also show that reproducible manner or intended to produce in systemic effect. And therapeutic effectiveness of a drug depend on the ability of a dosage form to deliver the medicament or to its sites of action at a rate and amount to sufficient elicit the desired pharmacological response. Means therapeutic response is also depend on the drug, or drug depend on the ability of a dosage form. Which dosage form you given to that particular patient for therapeutic effect, or the site of action of medicament, means which administration site is used for that particular administration of medicament and amount of sufficient to elicit the desired pharmacological response. That amount is sufficient or not means this therapeutic response is not only depend on single parameter, but is depend on different parameter like uh, dosage form and route of administration and amount that you administer or dose of the drug that you administer for pharmacological response. This attribute of a dosage form is referred to as physiological, biological, or simply viability. Hence, this is also show that the physiological ability or biological ability is simply viability of this form, and for most of the pharmacological response can be related directly to plasma level. Means you can also say that this effect of viability is directly related to plasma level, how much drug present in plasma to show the particular pharmacological response. So this is directly related to plasma level. Next, definition of viability according to different authors or different books. In this, according to viability by Goodman and Gilman, in this viability is a term used to indicate the fraction extent to which dose of a drug reaches to its site of action or biological fluid from the drug that access to site of action. It means this viability is the friction or amount of a drug to which drug can reach its site of action or to that particular action site to show the effect or biological food to access their site of action. And according to Brahmanian and Jaiswal, the term viability is defined as a rate and extent, that is amount of absorption of unchanged drug from its dosage form. Means how much drug is unchanged form or available in an unchanged form for excretion. This is according to Brahmanian and Jaiswal. Other definition is brand name. Brand name means first drug that is discovered and come in the market, and it's a branded drug. And the generic drug is the copy of that branded drug after completing the patent. And pharmaceutical equivalent means two drugs that are showing similar effect or similar ingredient or maybe same route or same dosage form, but manufacturer may be different. And according to definition, we can see that now brand name means a drug. A brand name drug is marketed under a proprietary trademark or protected name. Means it should be in a patent period or patent time and having a proprietary or right or trademark or protected name that nobody copy that particular name. And in the generic drug, a generic drug is same as the brand name drug in dosage, safety, and strength, and how it is taking quality, performance, and intended to use. These are the parameters which are maybe similar to a branded drug. And the pharmaceutical equivalent of the drug product are considered to be pharmaceutical equivalent if they contain the same active ingredient, have the same dosage form, and route of administration are identical in a strength or concentration. Means you can say that both are in the same active ingredient. In pharmaceutical equivalent drug, both drug having pharmaceutical active ingredient is same and same dosage form. Both drugs are applied by same route or same dosage form, and identical strength or concentration is also same. Means the capacity or the volume is also same. Those two drugs are known as pharmaceutical equivalent drug, and their manufacturer may be changed in this pharmaceutical equivalent, and brand name is under the trade name property or protected name, and generic is just copy of the branded drug. Next, pharmaceutical alternatives. In this pharmaceutical alternatives, these are the drug product that contain the same moiety and which contain different chemical forms such as ester or salt of the pharmaceutical alternative is somewhat also similar to pharmaceutical equivalent, but there's a little difference, that is innovator difference or maybe their moiety of a drug. Suppose one is using a normal drug or another is using modified form of the drug, that is salt form. So this is known as pharmaceutical alternative. Means these are the drug products that contain the same active moiety or same product but contain different chemical form. It's contain different chemical forms like product, ester form or normal ester form or salt form of the moiety, active moiety, but innovator may be differ or change that innovator product in dosage form and strength is also may be changed. In this condition known as pharmaceutical alternative. And Listed reference drug or reference listed drug is one book in which all drugs are listed, and these books are maybe standard or discovered or approved drug. And in this, you can check which drug are approved according to formulation of or for requirement of generic drug. In this, a reference listed drug is approved drug product to which new generic version are compared to show that they are bioequivalent. Means before preparing a generic drug or copying branded drug, you just check this branded drug or that particular branded drug, and according to their approval or their parameter which are approved, you can select any all parameter or these parameter to check both drug are equivalent or not or bioequivalent or not. Next is if size of a dose to be administered is the same, then viability of a drug is also depend on three major factors. Means if you are giving the drug by same dose or by same route, then in this condition it is show three major parameter or depend on three major parameter. One is pharmacological factor related to physiochemical property of a drug, characteristics of the drug. Second is patient related. Third one is route of administration. Means size, shape, all properties of the drug, and patient related, patient condition, if disease condition, any other like that he or she taking, which route you take to administer that particular drug. And within the parental route, intravenous injection of a drug result in 100% viability as the absorption process is bypassed. Means this route is show 100% viability when you given the drug by intravenous route, and drug is directly bypassed or absorption is directly bypassed due to intravenous injection and observe 100% viability. However, for reason of stability and convenience, most drugs are administered orally. Rather than parental route, some drug is also administered by oral route due to their stability, convenience problem. So, if you are giving the drug by parental route, they may require certain trained person, or it shows some stability problem or maybe convenience of administration. In this condition, to avoid this type of the problem, some drug is also given by oral route due to the stability and convenience problem. In such cases, the doses available to the patient called as bioavailable dose. Means you can also say that how much drug is available to that particular patient to show the desired effect. This is known as a bioavailable dose or available dose for that particular effect, and it is as an administered dose, and the amount of drug that reaches the systemic circulation, that is extreme of absorption, is called a systemic ability or simple bioavailability. Means how much drug is in systemic circulation or something available in systemic circulation. This is known as systemic availability or availability, simply availability. And the term bioavailable fraction refers to the fraction of administered dose that enter the systemic circulation. We already checked in previous slide that this viability is denoted by F symbol, or you use F symbol. In this, we can say that it's a formula for viability. F is equals to bioavailable dose divided by administered dose. And in this, to extend an optimal therapeutic action, active moiety should be delivered to its site of action. Means how much therapeutic effect is required or show the therapeutic effect, so that the delivery of the site of action is effective concentration for the desired period of a time. Next is objective of viability study. In this objective viability study, first is primary stage of development of a suitable dosage form of a drug entity to obtain evidence of its therapeutic utility. In this, you also known as a primary stage of development or suitable dose of development. Means you just confirm all your parameters are proper or not, or according to require or not, having sufficient capacity according to requirement or utility of therapeutic effect. And the next is determination of influence of excipients, patient-related factor, possible interaction with other drug on the efficacy of absorption. Means you can also check all excipients are compatible with drug or not, or maybe instead of drug, some excipient interaction is also takes place. So you can assure all excipients are in a good quality and compatible quality. Next is patient-related factor. Means if person is suffering certain disease, then it don't show any desirable side effect. don't show any side effect. It should produce required sufficient therapeutic effect or proper tablet disintegration or dissolution that takes place. Next is possible interaction with other drug or efficacy of the absorption. Means if you are formulating a two drug in a one formulation, in this condition you also check or confirm by this laboratory study that your two drug have a sufficient efficacy and no interaction is observed in this parameter. And development of a new formulation of existing drug, means if you are developing new drug formulation, so in this condition you use that previous study for the existing formulation, existing drug. Next is control of the quality of a drug product during early stage of marketing in order to determine the influence of processing factor, storage, stability on drug absorption. Means in this point you can say that you also check or confirm the quality control test or quality of your formulation. So if you just confirm by or maybe check parameter with the help of previous one branded drug, so it reduces certain stages or certain evaluation parameter, or you rightly assure that your formulation should be good quality or good storage and stability condition. Next is comparison of availability of a drug substance from a different dosage form or from the same dosage form produced by different manufacturer. Means you can also say that your formulation should be manufactured by same formulation or same manufacturer or by the different manufacturer. Means you just compare two drugs. In this comparison of viability of a drug substance from a different dosage form or same drug dosage form, means you are preparing two different dosage form. One is maybe liquid dosage form and second is a solid dosage form, suppose tablet and syrup. These two are different dosage form, but you are using same drug or a different manufacturer of both these formulation. Syrup is prepared by another manufacturer and tablet is prepared by another manufacturer. Thus both drugs are containing same dosage form. So in this condition, there is some similarity between this formulation. So you use this similarity for your formulation to finalize your formulation. In this next, significance of viability. In significance of viability, drug having a low therapeutic index, example cardiac glycosides, quinidine, phenytoin, antiarrhythmic, anti-diabetic, adrenal steroids, and theophylline. These are the drugs which have a low therapeutic index or low therapeutic effect. In this condition, you study your drug or check the viability of a drug. Example of these drugs is like cardiac glycoside or using drug for cardiac arrest, quinidine, phenytoin, and narrow margin safety of a drug. Also having example like antiarrhythmic, anti-diabetic, adrenal steroids, theophylline are the example of this narrow margin of the drug according to safety. Next is drug whose peak level are required for effect of the drug. Means you can say that drug whose peak level are required for effect the drug, that is drug like phenytoin, barbiturates, or bromidine, sodium valproate, antihypertensive, antidiabetic, and antibiotic. These are the examples whose peak level is required to show the effect of the drug. And these are the classes of the drug. Drugs that are absorbed by active transport. The example is amino acid analogs, purine analogs, etc. are the active transported drug. And in addition, any new formulation has to be tested for its viability profile. Means if you are formulating new drug or new formulation and you require to test the viability study or viability profile, in this condition you also require this viability study. Next is which drug which are disintegrated in the alimentary canal. Example is chromazine, etc. or those who are first pass metabolism. Means you can also check the viability of a digested drug or those drugs who are digested drug in alimentary canal or in a liver. In this example of this class drug is like chromazine, and these are metabolized by first pass effect. And next is formulation that gives sustained release of the drug. Formulation with a smaller disintegration time than the solution rate drug are replaced therapy also warranted viability testing. Means those drugs who are released in a sustained release or in controlled release have a small time to disintegrate or small disintegration time. In this condition, dissolution is also small time required, and so you can check this why replacement therapy or vulnerability testing is required. So in this condition or in this type of formulation, you also check this study by this viability. And the next is drug with strap dose response relationship, that is drug obeying zero order kinetics or mixed order kinetics. Example warfarin, phenytoin, digoxin, aspirin at a high doses, or phenylbutazine. And so you can say that when your drug having a mixed order kinetic, zero order as well as a first order or in a mixed order kinetic, in this condition you check the response in a proper way. So example of this class drug is like warfarin, phenytoin, digoxin, aspirin at a high dose you require, and phenylbutazone is also a class of this type of the drug. So you can check the viability by the different order, like it operates zero order or first order. Next is component of viability and bioequivalence. In this component, rate of absorption, the rapidity which a drug is absorbed. Rate of absorption means how much drug is absorbed in a particular period of time, or rapidity. Good pain. And slower onset of action is like a prolonged duration of action and to avoid side effects or adverse effect. Means rate of absorption, maybe you check that how much drug is show that particular may be available for that particular effect, or rapidity of a drug which is absorbed, and according to the rapidity we can classify in two types: rapid onset and slower action. Rapid onset means condition of like acute asthma or intense acute pain, we are required such type of drug. Means those drugs which are require emergency condition or require to leave or show the effect in within a seconds due to emergency condition like asthma attack or intense acute pain. So this is type of the drug is known as rapid onset. Second is slower onset. In this slower onset, if you require prolonged effect or prolonged duration of effect for long period of a time, or you avoid adverse effect, in this condition, slower action or slower onset of a drug is required. Next is extent of absorption. In this chronic condition like epilepsy, this is an extent condition of absorption. Next is different types of viability. In this first one is absolute viability, which is denoted by F. And according to definition, absolute viability is the when systemic ability of a drug administered orally is determined in comparison to its intravenous administration is called as absolute viability. Means you can say that viability or absolute viability is a systemic viability that you administered by oral route or which is administered by oral route, and when you compare this to intravenous route administration is known as absolute. Means you are giving a drug by oral route, and you just compare your this study with intravenous route drug administration, and in this percent absorption is equals to dose IV into AUC oral divided by dose oral into AUC IV. Means how much drug dose is given by IV route or area under curve for oral route, and divided by dose given by oral route or area under curve by IV route into 100. So after calculation you get percent absorption or absolute viability, and it is denoted by F. And a determination used to characterize a drug in net absorption property from extravascular site. Means you check your oral route property with IV route, and if you easily distinguish this ability by absolute value, then you can say that you compare your oral route with intravenous route. Next is relative viability, that is FR. In FR, when the systemic viability of a drug after oral administration is compared with oral standard of the same drug, such as aqueous or non-aqueous solution or suspension, if referred to as relative viability or comparative viability. Means it is also similar to absolute viability, and you just check two dosage form for the viability. But in this condition, there are little changes, that is use another formulation. Instead of changing of route of administration, you change your formulation in relative viability. If you are administered drug by oral route, and oral standard drug may be in a one solution form or maybe another suspension form or any other formulation, but you are giving in an oral route, or route of administration is same, but the dosage form may be changed. An example of this relative ability is comparison between the capsule of amoxicillin and suspension of amox. This is used to characterize absorption of the drug from its formulation. Means example is that you are taking the same drug capsule, but formulation is different. One is in a capsule form, another is suspension form, and you compare that absorption rate or the viability of this formulation. Formula for this relative viability, which is denoted by FR, is equals to AUC area under curve for A drug or AUC area under curve for B drug. Means one formulation and another formulation, ratio of one drug and second drug. Next is single dose versus multiple dose study. In this single dose versus multiple study, first you know about what is single dose. In single dose study, more common, easy and less tedious, less exposure to drug. You just take only single drug and you just start study. Or this study is more common and easy because you don't take any other drug. Single drug is used and less time taking and less exposure of drug is also in the single study. Next is disadvantages. In this advantage, it's difficult to predict study state characteristics. Means you don't check or maybe difficult or maybe show certain error during you check the study state characteristics. Next is multiple dose study. In this advantages of this multiple dose study is first accurate. You get accurate result by this multiple dose study instead of single dose study. Easy to predict the peak and valley characteristics. Means you can easily predict that peak, and few blood sample are required. Very less blood sample is required. Next is ethical. The small inter-subject variability. Variability is also small or inter-subject variability is observed. Better evaluation of control release formulation. Can detect non-linearity in pharmacokinetics. Means you can also check non-linearity parameter, can detect higher blood level, that is DT cumulative effect, eliminate the drug need for long washout period between the dose. And the disadvantages of this multiple dose is poor subject compliance, tedious time consuming, more drug exposure, more difficult and costly. Means more time is required instead of time, more cost or is also required, and difficult to perform because in this you compare multiple drugs, so there will be chances that difficulty to compare these drugs and continuous exposure or different exposure of the drug you suffer, and time is also required too much, and it's too much lengthy procedure. Otherwise, it gives overall good result or good parameters compared to single dose study. Next is human volunteer. In human volunteer, first is healthy volunteer versus patient you take. You just compare patient with your healthy volunteer, and in patient used in multiple dose study. And first point is patient get benefited from the study, reflects better therapeutic efficacy, absorption pattern, this is state evaluated, avoid ethical pattern. Means if you are taking patient in this condition, patient also have a benefit to overcome or maybe recover that particular disease. And instead of this, therapeutic efficacy is also better observed because you directly apply on the patient. And the absorption patient of disease state is also evaluated in good and proper way. And ethical quality is also complementary is also avoided. And disadvantage is disease state, other drug effect study. If person is suffering from a disease, so in this condition, you are giving any other drug, so that drug study may be affect and difficult to follow straight study condition. So you don't want to follow the straight state condition, but then certain or other errors are occur. Next is healthy human volunteers, and in this you check a drug effective or not in a human volunteer which are healthy, and in this you are taking young, healthy, or male or female, both type of healthy volunteer. If you are taking a female, then you make sure that you just sure that that particular female volunteer is not taking OC pills, and you study the OC pills. Body weight within a narrow range, and the body weight of that particular healthy person should be in a narrow range or according to requirement, restricted dietary, fixed activity condition. He or she don't take all type of food. There's a restriction in diet or certain restriction in diet, and a fixed quantity activity condition. He or she don't move in an unnecessary area or any other where the drug effect may be vary or show certain error. So in this condition, fixed activity condition, restricted dietary condition is also for healthy human volunteer. Next is pharmacokinetic and pharmacodynamic parameter. And in this pharmacokinetic parameter, it is divided into two classes, that is plasma level time study, and second is urinary study. Next is pharmacodynamic parameter. This is also known as direct method. In this, active pharmacological response, and second is therapeutic response. And in dynamic, you just check plasma level or urinary excretion data, because in this we already studied that pharmacokinetic is a ADM, that is absorption, distribution, and elimination. In dynamic means desired effect or therapeutic effect. In this, active pharmacological response, therapeutic response. In pharmacokinetic method, based on the assumption that pharmacokinetic profile elicit elected the therapeutic effect of the drug, and it shows the required sufficient therapeutic effect, pharmacokinetic effect of that particular drug. These are the indirect method, and in this you just check a drug effect or therapeutic effect in an indirect way. Next is pharmacodynamic method. It involves direct measurement of drug effect on direct measurement. This pharmacokinetic is an indirect method, and pharmacodynamic is the direct measurement. And in this pharmacokinetic, you just check the effectiveness of therapeutic effect of the drug in indirect way. But in this pharmacodynamic parameter, you check the pathophysiological condition of that person or function of that particular pathophysiological disease. And it is a direct method. You check the result or get result directly. In this first one, plasma level time study in pharmacokinetic parameter, the single dose study. This method is based on the assumption of the two dosage form that exhibit superimposable plasma time profile in a group of subjects should result in blood sample. In the single dose study, you can say that the blood sample sequence or series of this blood sample collection is required biological half-life, and these biological half-life may take 2 to 3. And after the administration, the analysis of drug are making a plot of concentration versus corresponding time. Means after a study of this biological half-life or 2 to 3 biological half-life, collect the blood sample with the help of this, and make a plot or graph, and check the analytical study. When you plot the graph, you plot the concentration versus corresponding time or required time. That is a collection of blood plasma level time profile, and with IV dose sampling does start with 5 minutes of the drug administration, and subsequent sample taken at 15 minutes interval. Means after administration of drug in IV route, the study gets started within a 5 minutes, and there's a sample collecting time at 15 minutes interval. Next is the 3 parameter of plasma level time study, which are considered important to determining viability are. One is AUC. AUC is proportional to the total amount of drug reaching the systemic circulation, and these characterize in this pharmacokinetic parameter is the total amount of drug that reaching in a systemic circulation. Next is Cmax, maximum concentration. It gives an indication whether the drug is sufficiently absorbed systemically to provide a therapeutic effect. Means how much drug is absorbed in the systemic circulation to provide a particular therapeutic response or therapeutic effect. And it is a function of both the rate and extent of absorption. And the Cmax will increase with increase in a dose as well as increase in absorption rate. This maximum concentration will be increased or should more increase effect when you increase the dose as well. Absorption rate is also increased. Next is Tmax. In Tmax reflect the rate of drug absorption and decrease the absorption rate. Means on Tmax or maximum time rate is reflect that it's just vice versa, and rate of absorption is decreased. When the rate of absorption is decreased, then in this condition absorption rate is increased. The extent of viability can be determined by following equation. F is equals to AUC oral D IV divided by AUC IV into D oral. And FR is the AUC test, D is standard divided by AUC test divided by standard. And so let us take two sample in absolute viability, that is oral or IV, because you are giving drug into different route, and the relative viability, that is FR, you taking the same drug with different formulation, that is test or standard drug, but is the dose of administered subscript IV and the oral indicate the route of administration, and subscript test standard indicate the test standard use of the same drug. Next is let's multiple dose administration. The methods involve drug administration for at least 5 biological half-life, a dosing interval equal to or greater than biological half-life, administration of at least 5 doses to each steady state, means in this will require 5 biological half-life. In previous one you require only 2 to 3, and you are giving a drug in 5 doses or check the steady state. And extent of viability, that is frictional viability, is AUC test into D standard or divided by test divided by into test and divided by AUC test, D standard and into standard, where T is dosing interval. Next is viability can also be determined from a peak plasma concentration at a steady state CSS max according to the following equation. Relative viability is equal to CSS max in test into D standard or omega T test divided by CSS max test D standard, and this is a graph. In this you can say that steady state is this, and area under curve is like this. Curve is known as area under curve. Methods to involve in this collection of a urine at regular interval of a time span equal to 7 biological half-life. You are taking a sample in a particular biological half-life or in a span to equal. Analysis of unchanged drug in collected sample. Determination of amount of drug excreted in each interval and cumulative excreted. And to obtaining the valid result of the following criteria must be followed. At each sample's collection, total emptying of the bladder is necessary to avoid the error. And if frequent sampling of a urine is also essential in the beginning, and friction exceed and change in the urine must be remain constant. Next is 3 major parameters examined in a urinary excretion data are as follows. dxu divided by dt max, and it gives the rate of appearance of the drug in the urine proportional to its concentration in systemic circulation, and its value increase at the rate of and the extent of absorption is increased, and Tmax is analog to plasma level, its value decrease as absorption rate increase, and X is related to a use of plasma level data and increase the extent of absorption and increase. This is plot of urinary excretion rate versus time. The extent of viability is calculated by the equation divided by x to infinity group IV or oral in this relative viability, absolute viability and relative viability. It's also ratio of x to infinity test into the standard divided by x to infinity into a standard into t test, means you just change the formulation test and standard instead of a root. With multiple dose study, this relative study viability is FR is equals to XUFS test into D standard into T test divided by XUFS standard into T test into T standard, and whereas the XUS is the amount of excreted unchanged during single interval steady state. Next is pharmacodynamic parameter. This is all about pharmacokinetic. In pharmacodynamic, first is active pharmacological response. In this use of pharmacokinetic method, chemical effects like change in ECG, EEG reading, and pupil diameter is observed in active pharmacological response, and the disadvantage is more variable. Metabolites interfere with results. Next, therapeutic response, second method. In this measurement of clinical response to the drug formulation given to patients suffering from a disease to which intended to be used, means how much therapeutic effect is observed that particular patient or suffering in that particular disease. In this disadvantage is improper qualification of observed response. By equivalent study are usually conducted using crossover design. Next is limitation of viability and bioequivalence study. In this limitation, the first is difficult for the drug for a long elimination half-life. It is difficult to those drugs who have a more half-life or long elimination half-life. Next is highly variable drug may require a far greater number of a subject. Then drugs that are administered by route rather than oral route dosage form are intended for a local effect, have a minimum systemic viability. Means if you are giving a drug by other route or instead of oral route, you are using another route, is difficult to check this viability study. Or if you are giving a drug in a high number, so in this condition also certain difficulty may be suffered for checking this. An example is like ophthalmic dermal intranasal inhalation drug product that you check for this viability study. By transformation of a drug make it difficult to evaluate the bioequivalence drug like stereoisomerism. Next is conclusion part after complete the study. Conclusion of this topic, that is viability is a key pharmacokinetic parameter which must be established for a new drug formulation or new modified administration. Many possible approaches exist to evaluate both rate and extent of viability, including situation for any intervention. Drug and study is not possible. These are the learning outcome after complete topic or completion of this viability and bioequivalence of this lecture. You should know about introduction of viability and bioequivalence, including object and significance. What is the object of the study and significance of the study, and complete introduction part or description of this viability and bioequivalence, and how to calculate viability and bioequivalence, means different method with their formula and component of viability and bioequivalence. And the next is measurement of viability that we already covered, pharmacokinetic and pharmacodynamic method. Pharmacokinetic is indirect method and it is further divided into two parts, whereas pharmacodynamic is known as direct method. It is also divided into two parts. We already covered this all topic in this lecture or in this PPT. Complete information and complete knowledge regarding the viability and bioequivalence. Next is reference part. In this reference, first reference is found in PL, Book and Elo, viability and its assessment, G-Vet Pharmacotherapy 27, 455 to 466 page number, Pharmaceutics and Pharmacokinetic Recipe by D1 Brahmankar, Sunil B. Jaiswal, second edition, Balla Prakashan, page number is C15 to 363. This is the reference. Both books are good and you can use both books for sufficient information. Next is question part. In question part, first question is pharmacokinetic is the option study of biological therapeutic effect of the drug. B is the study of absorption, distribution, metabolism, and excretion of the drug. C is the study of mechanism of the drug action. D is the study of the method of the new drug development. Second question, what does pharmacokinetics include? A is complication of the drug therapy. B is influence of the drug on metabolism process. C is drug biotransformation in the organism. D is influence of drug on gene. Next is, what does the term viability means? In this, A is plasma protein binding with the substance. B is the permeability to blood-brain barrier. C is the fraction of unchanged drug reaching the systemic circulation, and is the amount of substance in urine related to initial dose. Fourth question is the reason determining viability are. A is the rheological parameter of the blood. B is the amount of the substance obtained orally or quantity of intake. C is the extent of absorption and hepatic first pass effect. D is glomerular filtration rate. In fifth question, half-life is the time required to. A option is change the amount of the drug in plasma half-life during elimination. B is metabolize the half-life induced drug into half of introduced drug. D is final half of introduced drug in plasma protein. And in sixth question or next question, half-life does not depend on. A option is biotransformation. B is the time of the drug. C is the concentration of a drug in plasma. D is the rate of drug elimination. Next, seventh question is the rate of drug viability. is most rapid when the drug is formulated and its more viability is observed when you change your formulation or drug is formulated as is a controlled release product. B is a hard gelatin capsule. C is tablet. D is solution. Eighth question is if relative viability. We already discussed this relative viability. FR is one. It indicates A is viability of a dosage form. One drug is seen as that of the other dosage form. B is a complete binding of a drug to protein as compared to another standard drug. C is a complete viability of a drug. D is a complete distribution of the drug. In ninth question, the comparison of viability between two dosage forms is known as A is bioequivalence, B is viability, C is biopharmaceutics, D is biological. Means you just compare your viability study, like viability of two dosage form or two formulation, and after this study you check these two dosage form or two formulation, and this study is known as bioequivalence study, viability study, or biopharmaceutical study, or these biological study. Out of this four option ABCD, you select any one correct answer or correct option. Next, 10th question is viability of intravenous drug is always 100% by definition because A is viability measure the amount of substance that reaches in a bloodstream. B is absolute viability is 50% for any drug taken intravenously. C is absolute viability is much more important measure than relative viability. D is intravenous administration get the drug in bloodstream in fastest. So this is regarding the viability parameter. In this, you compare intravenous drug, check the 100% of your drug viability by taking the drug in intravenous route. And out of this four option, how can you measure or how can you check that which type of its viability it's given by that particular route? So you select any one correct option out of this ABCD. This is regarding all question part of this particular topic. After completing this viability by equivalence topic, we easily answer this question. So thank you so much for watching and listening this video. Thank you.